Hello, good afternoon, everyone. Let's get started. Let's begin with where we left off last week. We did not have a chance to do the application questions for lecture 7. So let's take them up first. So question number 1 here. You stare at a bright red screen for 30 seconds. What color will you see if the screen is switched to each of the following color? Okay, so if you're looking at the red screen for a long time and then you switch to a white screen, what color will you see? Okay, so red screen to white screen. Okay, what color will you see? Now, to answer this question, you have to look at the color wheel here, the primary color wheel here, which is in your lecture note. Okay, and so you have to understand your eyeball has 3 different types of cone cells in it. There is there are the blue ones, the red ones, and the green ones. And when you have a combination of the red cone cells and the blue cone cells sending signals to your brain, you interpret that as magenta, right? Just like a fancy word for pink, basically. And if you have blue and green being activated at the same time, then you see the color cyan. Between red and green, you would see the color yellow. Okay, and if all the red, blue, and greens are stimulated at the same time, then you would see the color white. Okay, so that's the first thing you need to understand. Now, sometimes you might stimulate the blue more than you stimulate the red, then you would get like a darker shade of magenta, right? You would see something like indigo or something. But if you activate more red than you do with green, then you might see something that's slightly more orange. So it's all about combinations of how much red is being stimulated and how much blue and how much green are being stimulated. Okay, so let's take a look at the situation. You are staring at a red screen and then you switch to a white screen. Let's try to understand what receptors do we need in order for us to see the color white? Now, if you were listening, based on what I told you, to see the color white, you would need the red receptor plus the blue plus the green, right? All 3 of them are going to be stimulated by the same intensity in order for you to perceive the color white. Okay? How about seeing red? Well, seeing red is quite simple. To see red, you only need to stimulate the red cone cells. So if you stare at a red screen for 30 seconds, for 1 minute, what happens is you will be stimulating the red cones. And when you switch to a white screen, the red cone cells, they get a little bit tired from the previous stimulation, right? So even though you're supposed to have the same intensity for all these cone cells, but the red one is kind of resting a little bit, right? Due to the previous stimulation when you were staring at the red screen. As a result, on a white screen, you would only be activating the blue and the green. And if you look at the color wheel here, blue and green actually gives you the color cyan. Okay, so if you stare at a red screen and you switch to a white screen, the white screen would give you a cyan color. Does that make sense to you guys? Any question about that? All right. Now why don't we maybe try this out and see if it actually work for you, okay? Give me a second here. Just trying to. Okay, so over here, I know you cannot see it. I have to find my buttons here first. All right, so there is a red screen. Okay, why don't you stare at this red screen? Okay. Oops, this is totally the wrong one. Sorry. Slideshow. Play on the primary one. Okay, let's try that again. All right, there we go. There is a red screen. Okay, so I want you to stare at the red screen right now. And what you're doing is you're stimulating the red cone cells. Okay, so they are transmitting the signal. They pick up the signal from the retina. They go through the optic nerve, past the optic chiasma, continue on the optic tract, and then the image would go to the thalamus, which ultimately will end up at your occipital lobe. Okay, so that's the pathway. That's the visual pathway, right? Now I'm gonna switch to a white screen. Okay, you blink your eyes a couple times. Okay. Do you guys see a light blue color, the cyan color that I was telling you about? Let me know in the chat box if you're seeing a light blue. Okay. So the reason, once again, you're seeing the light blue is the white screen is supposed, you need the red, you need the blue, and you need the green, right? But you tired out the red. Right? So only blue and green are sending signals to your brain, and blue plus green gives you the cyan color. All right, so I hope that makes sense to you. Now let's try a different question here. Oh, so the answer is cyan, okay, for part one. All right, next one. If you were staying at the color red and you switch to a green screen, red to green, okay, what's gonna happen if you go from red to green? What color do you think is gonna be? Let me know in the chat, or you can tell me directly. That works as well. All right, magenta, yellow, yellow. Okay. All right, let's talk about this. What what cone cells will be stimulated when you're staring at red? Just red, right? Okay. What about staring at green screen? If you're looking at a green screen, what cone cells do you need to stimulate? Remember, we only have three types of cones, right? Three types. We have the red, the blue, and the green. So which type of cone will be stimulated when you are looking at a green screen? Yes, it's green, right? So it's not a trick question. All right, so you stare in the red screen, your red cones get tired. You switch to a green screen. I don't need red cone cells to see the color green, right? So it's not going to get affected. It will still appear as green. Do you understand that? Right? It was it mattered. Excuse me. It matters in the previous situation because I actually needed the red cell for the second screen, right, to see the white. But here on the second screen, I don't need the red, right? So it doesn't matter that they're tired. All I need is a green cell, right? Okay, let's try the last one and see if it makes sense. You stare at the red screen and you switch to a yellow screen. All right. So to see the color red, you need the red. Okay, and now you need the cone cells, red cone cells. And if you switch to the yellow, what color will you see now? Okay, Tina says green. Anyone else? Rochelle also said green. All right, let's see if they are correct. To see the color yellow, right, I need red plus green. Red plus green. By the way, I want to clarify, right? These are light color, right? Like as in like visible light, right? They are not the same as like painting color. Right? Some people get really confused. They're like, well, if you mix red and green like paint, it actually becomes brown. Like I don't understand why it would be yellow. That's because we're talking about two different kind of colors. Okay, these are light color. The ones that you're thinking when you're painting, those are pigment colors, and they behave differently. In any case, if you want to see the yellow, you must need red and green. So if you tire out the red, you will only see green on the yellow screen. All right. I hope that makes sense. It's worth thinking about. I think there might be like at least one of these questions on the test. Any question before we move on? What is the answer? Oh, sorry. Green. Yeah, green. Yeah, because the red is out of commission. So you will see green on the yellow screen. Okay. How about orange? Oh, how do you see orange? I think that's what you mean. Orange is you would have to crank up the red a little bit. Right? If you stimulate the red more than the green, then you will see the color orange. Let me see if I can show you this. Give me 1 second, okay? I'm just gonna see where this thing is. Oh, okay, I found it. Okay, give me a second. Let me just share my screen with you. Okay, so you see over here, right? You can get something like this from like your paint or like um PowerPoint or any application basically. So like I have a shape here that's blue, right? And what you want to do is you want to pay attention to these colors, red, green, and blue. Okay. So let's say I set them all to like, 60 and 60 and 60, right? Okay, and then I'm just gonna turn up the brightness. Okay, so you see, oh, actually they're 250, 250, 250. If they're all stimulated it's equal amount, then it's going to be white, right? Just like I told you. Now, if I go to yellow, right? Think about the wheel again. If I go to yellow, which two numbers would be higher? Is it red and green, red and blue, or blue and green? Right? If I go to yellow, you can see. If I go to yellow right here, you can see there is not a lot of blue, right? It's just the red and the green, right? And that's what's going on inside your eye. Your red and green cone cells are being stimulated. Maybe a little bit of blue stimulation, but majority is red and green, right? So to see the orange color, right, you would have more red stimulation than green, and then again almost no blue, right? So all the colors that we see are just combinations of how much of the red, how much of the green, and how much of the blue cone cells are being stimulated. Does that answer your question, Hima? Excellent. Thank you. So let's go back to our study guide here. Question number 2. This was quite simple. David went to his yearly eye examination and was informed that his intraocular pressure, okay, sounds like a big word, but that just means the pressure inside his eyes, was slightly elevated. The physician expressed concern over his condition and noted that if the condition got worse, eye drops would be needed. What is wrong with his eyes and what are possible consequences of this condition? Speculate the function of the eye drop, include correct and relevant eye anatomy in your answer. Okay, so based on this description, what is the condition that the doctor is worried about for David's eye? That's right, glaucoma. Okay, so glaucoma. And I explained to you that glaucoma is essentially high eye pressure at the back of the eye, which would then compress the optic nerve, right, which cause you to lose vision progressively. All right, there are no treatments for glaucoma, and the eye drops are there to help reduce the eye pressure. Okay, so the eye drops could achieve this in a couple of ways, right? It could increase the drainage of the fluid in the eye. Right? It could slow the production of the fluid, like the vitreous humor, for example, or it can like relax the eye muscle, so that we could lower the eye blood pressure, eye pressure, not blood pressure, the eye pressure. All right, so that's it. That's basically everything in lecture 7. Let's move on to lecture 8 now. This is probably much easier compared to the previous 3 lecture that you did, right? Like with the brain stuff and everything, that's just like a little bit overwhelming at times. So hopefully this is a little bit of reprise for some of you. Let's go through it, and then we will take up the lab, and then I'll send you on your way. First question, list 4 accessory organs in the digestive system. So just to recap, the GI tract starts at the mouth, right? The mouth. And then it's going to go through all these stuff in between. We're going to have, esophagus, stomach, so on and so forth. And it's going to come out on the other end, the anus. Right? This whole tube is a continuous tube, and we call this the alimentary canal. Alimentary canal. Okay, or GI tract. Okay, or GI tract. Along the way, there are accessory organs that would add various mixture to the GI tract, right, to help with the digestion process. The food themselves do not go into the accessory organs, right, but we would need the accessory organs as part of our digestive process. So let's go through some examples of accessory organs. At the beginning, in the mouth, we have the salivary gland. Salivary gland. Now, there are 3 pairs of salivary gland, right? Does anybody want to tell me what they are, or you just want me to write it? If you know what they are, please tell me. 3 pairs of salivary glands. Starting with the one next to a ear. Yeah, Nor said. Yep, sorry, go ahead.
Sublingual.
Yep. And which one is that? Where is it? Do you remember sublingual? Where might that be?
Below to the tongue.
That's right. Yeah, lingual means tongue, right? So sublingual, very good. And then there's one last one. Parotid is next to the ear. That's correct. And the other one is Hima said is submandibular. Yeah, I think that's what you meant, but close enough. Mandibular. Okay, this is called the mandible, your lower jaw. Okay, so like over here, that's where the submandibular salivary glands are going to be. Okay. Next, another accessory organ, we have gallbladder. Gallbladder. Now, you can actually live without your gallbladder. The function of gallbladder is to store the bile, right? That is being produced by the liver. Right? So liver is another accessory organ. You cannot live without your liver. And one last one. Anybody remember what the last one might be?
Pancreas.
Pancreas is correct. Thank you. Pancreas. Okay. You can live without your pancreas, but you will have to be on a lot of medication to replace all the enzymes that it normally would produce. Okay, so those are your accessory organs. Now your alimentary canal, it's essentially like a long tube, right? And the tube is made up of 4 layers of various connective tissue. So starting at the most superficial, the outermost layer, we have the serosa. Okay, this is outermost layer. Okay, the most superficial. We use correct anatomical term. Most superficial. Okay, outermost layer. Beneath the serosa is going to be the muscularis. Okay, muscularis. This is the layer that has smooth muscle, right? That helps, squeeze and push the food forward along the alimentary canal. Now, generally speaking, the muscularis has 2 layers of smooth muscle. It has the circular, which is responsible for the squeezing action. And then there's a longitudinal, which is responsible for the pushing action. Now, the stomach has an extra layer. Everywhere else along the GI tract, there are only 2 layers, the circular and the longitudinal. But in the stomach, there's an additional layer called the oblique layer. which gives you like twisting motion. Okay. Beneath the muscularis is the submucosa. And this is where you will find things like blood vessels, right? Nerve endings. And it's where you will supply nutrients, oxygen, and all that stuff to your GI tract, but also it will help you absorb the nutrients from your food, right? Specifically at the latter two parts of your small intestine. And finally, the layer that is the deepest, that is in direct contact with your food, that is the mucosa. Mucosa. All right. Next, along your GI tract, we want to keep things moving forward, okay? It's a disaster and it doesn't feel good every time things start to go backward, right? If you ever vomited, you will know it's not a good feeling, right? For food to come out, right? So that holds true along the entire way. We don't want stuff in the large intestine to start going back to the small intestine, for example. Okay? Everything should be going forward, and to keep it that way, we have these sphincters along the way, right, to prevent backflow. Right? You can think of sphincters sort of like valves in the heart, okay, and the blood vessels. So 4 major sphincter. The first one that is on the between the esophagus and the stomach, that is the cardiac sphincter. Cardiac sphincter. Now, in the notes, I believe this is also called the lower esophageal sphincter. I like cardiac sphincter more because you know, it helps you remember when a person gets like a heartburn, it is because the cardiac sphincter is it's not closing properly. So all the acids start to come out from the stomach, and it burns your esophagus a little bit. Okay, so this is the cardiac sphincter right here at the top of the stomach, and this is where all the acid is, HCl. And down here, that's the second one. It helps regulate how much of the stomach content will go forward. Does anybody remember what the name of the second one is? Thank you, Nora. This is the pyloric sphincter. Okay. And then between the last part of your small intestine, and just for the record, the small intestine is separated into the duodenum. That's the first part. The jejunum, and then the ileum. Right, so the last part, the ileum, connects to the cecum, which is the first part of your large intestine. And there's a sphincter that separates the two. We call it the ileocecal sphincter. Okay. And then finally, there is the internal and external anal sphincter. Okay, that's basically its final control, right? Before you let the stuff comes out. Okay, so one of them, the external one, is under voluntary control. Internal one is not. Okay. So those are the sphincters. Next, the chemical digestion of protein first begins at. Okay, so where do we first begin the chemical breakdown of proteins? Does anybody remember from the lecture? Is it the mouth? Is it the stomach? Is it the small intestine? Where do we start breaking down the proteins? Okay, Nora said mouth. Anyone else? Do you agree with her? Or stomach, Tina, stomach, Maphis? Yeah, stomach is correct. Okay, stomach. Okay. In the stomach, that's where you will first expose the protein to pepsin. Okay, so pepsin in the stomach breaks down protein into amino acid. Okay, not necessarily like individual amino acids. It could be like chains of amino acid, like shorter chain, but that's where the chemical digestion first begin. Where we don't have enzymes in the mouth, in the saliva, that will break down the protein. But in the mouth, we do have enzymes for breaking down carbohydrates. Right? We have amylase in the saliva. So chemical digestion of carbohydrates begins at the mouth. As for lipids, you don't start breaking them down chemically until you get to the duodenum. Okay, again, duodenum, for those of you who forgot, duodenum is the first part. First part of small intestine. Okay, it's the shortest part, about 12 inches long. All right, next, state a function of each of the following enzymes. So there are a lot of enzymes in the body, in the digestive tract, that breaks down various macromolecules, and much of these enzymes are going to be found in the pancreatic juice, okay? The secretion that is made by the pancreas. And here are some of them. Okay, so amylase, you will find it in saliva. You know, I'm just gonna write down found in saliva and pancreatic juice. Okay. And what does it do? Well, as the name implies, the amylase is going to take amylose, right, and break it down into glucose. Plus, again, it doesn't have to be like individual glucose. You might not have enough time. The carbohydrate might not spend enough time in the mouth to completely get broken down into individual glucose. So it might just be oligosaccharide. Right, little short chains of sugars. Right, so that's what amylase does. Now, pepsin and trypsin, they are like cousins. They're related in terms of, you know, functionality. And the function of pepsin and trypsin is to take protein, and then it will chop it up into amino acid, okay, or short strands of amino acid. Okay, think of a protein as really long like that, okay? So what happens is you will chop it up. You will chop, you might chop one up, you might chop here, you might chop here, chop. So in the end, you will have individuals amino acids, but then you might still have some short chains. Okay, so this process begins in the stomach, and then it will get finished up in the duodenum. Okay. Last one here, lipase. Lipase is also found in the pancreatic juice, and it basically takes your lipids and it will break it down into glycerol plus fatty acids, right? You may remember learning about these last semester, right? So that is that. Okay. Next, list 3 functions of hydrochloric acid in the stomach. Okay, so hydrochloric acid, one of the functions is to denature proteins. And you unravel it, unfold it, right? And by denaturing the protein, it makes it easier for pepsin to break it down. Okay. Second function of hydrochloric acid is to activate pepsinogen into pepsin. Okay. So when you produce the pepsinogen, it is inactive, right? And you must turn it on into the active form, and that will be the function of HCl. Does anybody remember what produce pepsinogen in the stomach? You have different cell types, right? Which one is responsible for producing the pepsinogen? That's correct, Nor. That's the chief cells. Okay, so pepsinogen is produced by chief cells. Okay, whereas the hydrochloric acid, that's produced by parietal cells. Okay. And one last function for hydrochloric acid, basically immune defense, right? Okay. It's part of your chemical barrier. Number 7, list 3 features that increase surface area in the small intestine. All right, so first you have these circular folds. Right, so if you think about your small intestine, if you take a cross section of it, it's not smooth on the inside, but you know, it has all these circular folds. Okay, so if you take one of these circular folds and you zoom in into it, you will see there are additional foldings. Okay, so these are called villi. Okay, villus is singular. And if you take one of these things out and you look at the type of cells that makes them up, you will have some cell here. Okay. And then if you zoom in onto the cell, you will see these cells also have little hair looking things. Okay. Those are called microvilli. Okay, so collectively, all these extra foldings, they're going to increase the surface area substantially. Okay. They increase it from a total surface area of about 400 to over 2,000 square feet. Okay. And it's important to have large surface area in the small intestine because it gives you more room for absorption. It gives you more room for reactions to take place. Okay. So collectively, it would increase the efficiency of the function of the small intestine. All right, let's do some true or false. First question, chemical digestion of food involves digestive enzymes that links monomers to form polymers using hydrolysis. This is not true. Not true. Enzymes, they do not link monomers, okay? So they break polymers such as protein, carbohydrates, into monomers. Okay. The process is called hydrolysis, though. So that's not wrong. This part is okay. But this is where the mistake is. Pepsin is an example of an enzyme that breaks down protein. That is not that is correct. That is correct. Okay. Pepsin is found in the stomach, as well as the pancreatic juice, right? And we just talked about it. The pepsin is made in the form of pepsinogen and must be activated by hydrochloric acid before it is going to be active. Amylase is an example of an enzyme that breaks down carbohydrate in the mouth. That is correct. The liver is an accessory organ that secretes bile into the jejunum that helps in lipid digestion. That is not correct. First of all, it is not the liver that secretes the bile. Well, the liver makes the bile, but then it's stored in the gallbladder, right? Okay, so from the gallbladder, the bile is going to be secreted into the duodenum. Okay, and we need the bile for lipid digestion. This is false.
Professor,
Yes, please.
before you said chemical digestion starts in stomach, right? Up there. And then the question number 3 says amylase is an example of enzyme that breaks down carbohydrate in mouth. Isn't it the chemical digestion then?
Okay, so the protein,
Yeah. Yeah.
right? It's the stomach, right? And the mouth is the
Oh,
carbohydrate, right? Yeah,
okay. Thank you.
no problem. Where am I? Okay, number 5. The epiglottis prevents food from entering the nasal cavity during swallowing. This is false. What is wrong with it? Does anybody want to tell me what I need to change?
The epiglottis helps to prevent entering to the trachea, not the nasal cavity.
Yeah, that's correct. Okay, so the epiglottis prevents food from entering the trachea. Okay. Alternatively, if you want to keep the nasal cavity, this would have to be uvula, right? Okay. Uvula is the thing that dangles at the back of your throat that prevents the food from going up into the nasal cavity. But you know, it's okay. We can stick with this correction. Next one, different parts of the tongue is responsible for detecting a specific taste. That's not correct. You've probably seen a diagram where, you know, this is the tongue and they say, oh, that's like salty, that's like for sweet, and bitter is at the back. But it turns out the whole tongue is sensitive to all the different tastes. Okay. Then the taste, right? Just to review for you, it is sweet, sour, bitter, salty, and then the last one is the umami, right? Which tastes, that gives you like a savory taste that you get when you eat things that contains MSG. The pyloric sphincter prevents acid reflux. That's not true. It is the cardiac sphincter. Food is pushed down the esophagus by wave-like contraction called peristalsis. This is an involuntary contraction of smooth muscle. That's true. Okay, so the peristalsis is carried out by the muscles in the muscularis layer, and it's going to continue throughout your GI tract. Small intestine is separated into 3 regions: colon, rectum, and anus. That would be the large intestine. Small intestine is separated into duodenum, jejunum, and the last one is the ileum. chyme is neutralized by pancreatic juices in the stomach. That is false. Okay, the chyme is neutralized by pancreatic juices in the duodenum. The food is still, being digested by the acid in the stomach. No point neutralizing it there. By the way, what is in the pancreatic juice that helps you neutralize the chyme? What's the chemical that's responsible for doing that? Sodium bicarbonate, okay? That's in your notes. Duodenum is mainly responsible for nutrient absorption, while jejunum and ileum is responsible for nutrient digestion. It should be the other way around. Okay, it's the other way around. You have to digest the food first before you can absorb it, right? Bile works by emulsifying fat which aids in their digestion. That's true. To emulsify them just means it helps them bind with water. It's like your detergent, right? If you try to wash your greasy dish with no detergent, it's really hard because the fat won't bind to the water and you can't wash them off. But when you add the detergent, it emulsifies the fat. It allows it to bind to the water and it will wash off the plate along with the water. Villi are found in small intestine to increase surface area for nutrient absorption. That's true. We did that. All right. Let's go through the fill in the blanks here. The tube that connects, that word shouldn't be here. The tube that begins at the mouth and ends at the anus is called the alimentary canal. The innermost layer of the alimentary canal that is in direct contact with the food, mucosa. The layer of alimentary canal, sorry with all these mistakes, that contains blood vessels, lymphatic vessels, and nerve is called the submucosa. If you push up with your tongue in your mouth, you will feel the hard palate. Okay, so that hard palate is formed by two bones, the maxilla at the front and the palatine at the back. The maxilla actually also form the upper part of your mouth, right? That's where your upper row of teeth are inserted into. The uvula is an extension of the soft palate, which prevents the food from going up to your nose when you swallow. The fancy word for chewing is called mastication. Okay, you have two muscle that helps you do that. Your temporalis. If you put your hand in the temple here and you chew, right? You pretend you're chewing, you will feel the muscle moving. And there are two additional muscle here, masseter, that helps move your jaw. And we'll learn that in the muscle unit. The gland that is located next to the ear, that would be the parotid salivary gland. Enzyme found in saliva, which breaks down amylose, that would be amylase. A bolus is a ball of food. Peristalsis are wave-like contractions along the GI tract that propels food forward. 3 layers of muscularis in the stomach. Okay, so it has the same 2 layer as everywhere else in the GI tract, the longitudinal and the circular. But there is an additional one that I told you about, the oblique. diagonal pattern that allows twisting of motion. Folds found in stomach lining when it is empty, those are the rugae. Okay? Like an empty balloon, there are wrinkles. That's basically what it is. But the rugae will disappear when your stomach is full. A hole in the diaphragm where the esophagus passes through, that is the esophageal. Esophageal hiatus. Okay. Esophageal hiatus. Sorry. There we go. Thick, acidic, soapy liquid that leaves the stomach. That's the chyme. Okay, so when the food has been sitting in your stomach for a couple of hours, the acid has break down much of it, and it becomes these soapy mixture called the chyme. Pepsinogen is activated by HCl into pepsin. You know that by now. The parietal cells produce the HCl, but it also produces something called the intrinsic factor. It's a sugary protein, a glycoprotein, that will bind to B12 so that it's not going to be broken down by the acid. Okay, we need the B12 to make healthy red cells. And if you don't have enough B12, then you might become anemic. The three parts of small intestine are duodenum, followed by jejunum and ileum. Lipids, because they don't mix well with the liquid part of the blood, they must be absorbed through special things called lacteal in the small intestine. They are taken to the liver where they will be combined with lipid-carrying proteins before they can enter the bloodstream. Bile causes the lipids to break down into smaller droplets in a process called emulsification. When you are starving, the liver can convert glycerol into glucose in a process called gluconeogenesis. Okay. The new creation of glucose. And then we have the NaHCO3, the sodium bicarbonate. That is the buffering component of the pancreatic juice. Okay. That's it for the fill in the blanks. These are very straightforward questions. Does anybody have any requests for any of them? Number 6. Sure. So pancreatic juices basically has two major components as far as we are concerned. There is the NaHCO3, which will help neutralize chyme in duodenum. The stomach is not going to be affected by the acid because it has a thick mucus lining around it. But when the chyme enters the duodenum, there are no mucus lining in the duodenum. And so you have to neutralize these chyme. Otherwise, it would start to irritate your small intestine, right? And that's what the sodium bicarbonate is going to do. It's basically a chemical reaction, right? The hydrochloric acid You probably learned this in chemistry, plus NaHCl3 is going to produce NaCl, so salt, plus H2O and CO2. Right? So digestion creates a little bit of gas. People just burp it out usually. And in addition to the NaHCl3, there is a mixture of digestive enzymes. Right? There are like 28 of them at least, but we just have to know the basic ones, right? Like amylase that we talk about. There's the pepsin, there's the trypsin, right? Lipase. So all these digestive enzymes will also be secreted into the duodenum, and they will work together to basically break all the macromolecules that are in our food into the monomers, so that we can absorb them later on in the jejunum and the ileum. Okay. All right. A patient has esophageal cancer and must have a feeding tube inserted. The nurse tells the patient that the tube will be inserted surgically into the duodenum. The patient's wife asks why the tube will not be inserted into the stomach. What should the nurse say? Okay. Why are we inserting directly into the duodenum and not the stomach? What do you guys think? Okay. So when patients are on a feeding tube, the solution that or the food that you feed them is not solid food, right? Okay, they're in a semi-liquid state, right? So it's already quite processed, quite broken down, right? That's why there's no need to go into the stomach, okay? You can feed the solution directly into the duodenum, and then let the enzymes from the pancreas to continue the breakdown. Okay, because again, we're not feeding them like solid chunks of food. There's no need to, go through the acid phase of the digestion. Okay, so if you imagine like eating, steaks or eating a very clunky stuff, like even carrots and stuff like that, right? They're really big chunks, and the acid helps break it down into a soapy mixture, right? But when you give the food to a patient with a feeding tube, it's already in a semi-liquid state, right? It's basically like baby food, right, so to speak. And that's why you don't need to go through the stomach. And a second reason is you don't want to be inserting any kind of tubes into the stomach unless you absolutely have to, because the stomach is a very acidic environment. It will cause erosion of whatever thing you're trying to insert into the stomach, right? And you don't want the stuff to be leaking out from the stomach either, right? Which could irritate your abdominal cavity. So that's the main reason. Okay, you just ate a large poutine. Use correct terminology and anatomy. Describe how poutine will be digested and absorbed in your digestive system. Pretty straightforward question. For those of you who don't know what a poutine is, it's basically this French fries things with gravy on it. So, what kind of macromolecules do we have when you eat something like that?
It's a high carbohydrate.
Yes, so there's a lot of carbohydrates from the fries. Okay, and that would, the digestion of that would begin in the mouth and continue on in the duodenum, right? So this is from the fries. Okay, what else? What other lipid? That's right. Where is the lipid from? Do you guys know? Also probably from the fries, because you fry the fries in oil. But what else? Yeah, the gravy, right? The gravy is all fat. I'm telling you, It might taste good, but it's gonna clog your artery really good. Okay, so all these lipids are gonna not gonna be digested until you get all the way to the duodenum, right? So you will have this big glob of lipid, and then you would have the bile from the gallbladder that makes it into little droplets, right? And then you would have the lipase from the pancreatic juice that would actually break it down into, fatty acids and glycerol, right? Okay, so these are fatty acids. So it's a two-step process. And this might be surprising to some of you, but there's one more thing here that we can add. What is that? Okay, the protein. Where is the protein from in the poutine? The cheese, that's right, the cheese. Okay, cheese is primarily protein and fats. And as for the protein digestions, it would happen first of all in the stomach, and it will continue on to be broken down further in the duodenum. Okay, so that's what it is. Okay, last one. Chronic inflammation of the pancreas or pancreatitis impairs the digestive function of this organ. What would be the effect on digestion and absorption of these macromolecules? Well, the pancreas is again responsible for secreting the NaHCO3 plus the enzymes, right? Okay, so basically, if you are missing all the enzymes, right? If you have chronic inflammation and the output of enzyme is affected, you will have trouble completely digesting these things. Carbohydrates might be okay, and proteins might be okay because you do a little bit of digestion of carbohydrates in the mouth, but not nearly enough, right? Think about how long the food stays in your mouth before it gets swallowed, right? Like we don't really chew our food too long. Okay, like maybe like 15 seconds, 20 seconds, and then you swallow it, right? So the amount of carbohydrate digestion that happens in the mouth is very limited. Okay, especially if we're talking about like complex carbohydrates, right? For simple ones, like if you're eating candy or if you're chewing on some slice of bread or something, those are like simpler carbohydrates that you can maybe start to break down quite a bit. But if we're talking about complex carbohydrates like those found in like grains, quinoa, that kind of stuff, and then really there isn't much digestion going on in the mouth, and without the enzymes from the pancreas, you would really have trouble breaking them down, right? Same thing with the fats, right? The the bile will make it into smaller droplets, but you really need the lipase from the pancreatic juice to finish the job. Okay, so in general, missing a bunch of enzymes is going to essentially result in indigestions. Okay, you're not gonna if you cannot break it down into very small monomers, then you are not going to be able to absorb them as nutrients. So you will basically suffer from malnutrition as well, right? You might start to lose weight, and overall it's just not good. Okay, but there are like enzyme supplements that you can take, like a mixed pill, that people can take if that's the problem, and it would kind of help solve the problem. Another thing is without the NaHCO3, you will irritate your duodenum, right? Because there's nothing neutralizing the chyme. And if you keep on irritating your duodenum, it might even burn through the mucosa and the submucosa, resulting in duodenal ulcer. Okay, so it's not a good thing, but that's basically what it is. Okay. All right, let's maybe do a few polls, and then we'll take a break. And then when we come back, we will do the lab. Okay. So first question. There's the poll. Okay, thank you. Everyone who answered got it right. The appendix is a little pouch that comes out from the cecum, and that is not an accessory organ. It doesn't really do anything. You can live without an appendix. Okay, number 2 here. Okay, most of you got it right. It's the submucosa. The mucosa is the layer that's touching the food. No blood vessels, lymphatic vessel there. Muscularis is where you will have the smooth muscle that helps propel the food. Okay, so blood vessels, lymphatic vessels, nerve endings, that's going to be in the submucosa. Okay, let's try a few more. Number 3. Yep, it's the one next to the ear. It's actually labeled right here. What is the function of amylase? We talked about that multiple times already, is to break down carbohydrates. So I'm not gonna ask you to do that, but let's do number 5, please. Number 5. Okay. Because there's a lot of different things going down your esophagus, right? Hot stuff, cold stuff, spicy stuff, right? Acidic stuff. All these things gonna irritate the esophagus if we don't have a good lining for protection. Okay, and you learn from the last semester the type of epithelial tissue that gives the most protection against these extreme conditions would be the stratified squamous, multiple layers of flat cells. So that's the answer. Hydrochloric acid is produced by parietal cells. We talked about that earlier. And I'm just going to take up the other ones with you. The function of bile is to chemically break down fat. That's not it. Okay, that would be the function of lipase. Okay, physically break down fat. That's what it does. The emulsification is to break it down into smaller droplets. To absorb fat into the bloodstream, that's the function of lacteal. And there's no such thing as reduced fat absorption. Last but not least, the purpose of sodium bicarbonate in pancreatic juice is to neutralize the chyme in the duodenum. Okay, so there is that. Now, before I take a let you take a break, let me just go to the link for the lab. I'll post the link to lab 2 in the chat box. Okay, so you can download a copy, and then we'll take it up afterwards. All right, so I'll see you in 5 minutes. Okay, so hopefully you've got a copy of the lab in front of you, and we're gonna take it up right now. Let's take a look at the first slide here. Okay, so this is the brain, and we're looking at it from the side. That's why it's called a lateral view. Are we looking at the right hemisphere here or the left hemisphere? Anybody tell me which half of the brain we're looking at? The left half or the right half? Okay.
Looks like right.
Yeah, so this is the front, right? This is the front. You can tell this is the front because this is the cerebellum, right? Cerebellum is at the back. Okay, and this is the lateral surface, so the outside. And if this is the front, this is the back, then this must be the right hemisphere. Okay, right hemisphere. Okay. All right. Identify the lobes A, B, C, and D. Okay, so lobe A, I mean it's the boundaries are not really clear here, but let me see what's a bright color. Maybe that. Okay, so here is A. This whole lobe is A, right? Okay, so A is the frontal lobe. B is over here at the back. Okay, so that would be the parietal lobe. C is located at the back. That's the occipital lobe. And D would be the temporal. What sensory information are processed by B, C, and D? So for B right here, there is the somatosensory cortex. So that would be responsible for processing somatosensory inputs. Okay? And if you forgot what that means, it's like sensation, things that you can feel with your skin, right? So touch, texture, temperature, all that thing, pain, all those things are somatosensory inputs. For C, it is occipital lobe, so that is visual input. And for temporal, it is auditory, which is sound, and olfactory. Okay, so smell and sound. That is what they will process. Okay, E, F, and G. E is the cerebellum, located at the back. And what kind of memory are stored at E? Stores skill memory, right? So typing, right? Riding a bike, shooting a basketball. That's skill memory and it's stored in the cerebellum. F, anybody know what F is? This whole thing is the brain stem, right? Yeah, that's correct. F is the pons, which means bridge, like in French, right? Bridge is pontus, I think. So that's the bridge that connects different parts of the brain together. And in G, we have the medulla oblongata. And that's where you will have your respiratory center. It's where the tracks are going to cross the midline, for example. Let me know if I'm going too fast, okay? I can slow down a little bit. Label the functional cortex in H and I. Now you should know that this right here, there is a groove. A groove is called a sulcus, right? So this groove separates the frontal lobe from the parietal lobe. That's called the central sulcus. Okay. So in front of the central sulcus, you have I, which is the primary primary motor cortex. Okay. And the function of the primary motor cortex is to allow voluntary movement of skeletal muscle. Right? Not every muscle is under voluntary control. Right? The muscle in your GI tract, for example, is under autonomic regulation. Right? So you cannot physically control those. So anything that can move voluntarily in the body, that starts in the primary motor cortex. Okay. Behind the central sulcus, you would have the primary somatosensory cortex, which, as I mentioned on the previous slide, will process somatosensory inputs, such as touch, pressure, temperature, texture, pain, anything you can feel with your skin. Okay, this is a medial view now. You're looking at it from the middle, and anytime you see a brain from the middle, you will have this thing, right? I mentioned about this, and that would be the corpus callosum. It's white matter, okay, and it allows communication between the two hemispheres. Okay, that's corpus callosum. B is thalamus. Okay, so thalamus is responsible for filters sensory information. Before passing them to the appropriate lobe. Okay, so all the sensory information passes through the thalamus except for smell. Okay, smell, your nose is right here. It doesn't make sense to go up there and then come back out to the temporal lobe. It just goes directly to the temporal lobe. Okay. Label C and D. Okay, C is so again, this is the thalamus. You can always tell the thalamus because there is the oval with the dot in the inside. It's almost like an eye, okay? And then below that, all right, is the hypothalamus. It literally means below the thalamus. Okay? So this is the master gland. Master gland. It regulates a lot of things through hormone. Maintains homeostasis and internal balance, right? Through hormones. Okay. So salt balance in the body, your metabolism, water balance, sleep wake cycle, everything that I've said is regulated by the hypothalamus, either directly or indirectly. So thalamus, hypothalamus, and then there's a thing that dangles below the hypothalamus, which would be the pituitary gland. So this uses hormone to regulate various processes in the body, right? So actually these two things work very close together, and we'll learn it in lecture 12. Right? Both of them plays a role in homeostasis and regulation through the use of hormones. Is everybody okay? Like, is this pace all right? I hope I'm not going too fast. Yeah, okay, thank you. All right, and other thing here. Now this is maybe a little bit hard to see. We did this 2 weeks ago, I think. Yeah. So I'm just gonna go back a little bit first, huh? Let me choose a good color to. It's really hard to highlight on these things. Okay, so again, corpus callosum, this band, and underneath the corpus callosum there is a gap, right? I told you about this gap. If you stick your hand in, it opens up into a wide a chamber, which would be the lateral ventricle, right? The lateral ventricle. From there, it's going to come out in front of the thalamus, which would be the third ventricle. Okay, and then it goes down through the cerebral aqueduct, which would then bring you to this triangle structure. So this chamber between the pons and the cerebellum, that is the fourth ventricle. Okay, that's the fourth ventricle. And the G here, that is basically the central canal. Right? And that goes down all the way down your spinal cord, right? Okay. All right, more brains. This is a CT scan. On the CT scan, identify the following. Okay, so start with the corpus callosum. Hopefully that's the easiest one for you to see now. Okay, corpus callosum is this guy right here. You will always see it in a medial view. Cerebellum right here at the back. Right, it kind of shows you that these things are real, right? Like I didn't make them up. You can see it in the CT scan. The pons, that's this thing right here. Right, and you can see the fourth ventricle between the pons and the cerebellum, right? And the medulla oblongata. Medulla oblongata is right here. Okay. Hopefully you saw that as well. All right, more things here. Lateral ventricle. That's right here, right? At least that's part of it anyways. Remember the whole thing, it's like a C-shaped horn like that, right? Very, very big. But from a medial view, we can only see a little bit of it right underneath the corpus callosum. Third ventricle is right here in front of the thalamus. Right? It's like, you know, you have a thalamus here and a thalamus here, and the gap between it, right? That's where your third ventricle is going to be. Cerebral aqueduct right here, right, which connects the third to the fourth. The fourth is a triangle, right? Okay. It's good to just, like, you know, Google brain diagram or brain image, and just start to identify things when you're studying. Right? It's a good way to see different structures from different diagrams. That helps you understand it a little bit better. Okay, this is another brain. You're not gonna believe it, but this brain costs $400 when when I purchased it 5-6 years ago for the lab, and it's super flimsy, instant buyer regret. In any case, you will not get to handle it. We just have to look at it under the in the diagram. So list 3 functions of A. A is this thing right here. Okay, and hopefully you know by now that is the cerebellum. So the cerebellum maintains balance and equilibrium. Stores skill memory, enable smooth execution of voluntary movements. Okay. Label B. Okay, this here. It's hard to. Oh, you want to go back? Yeah, I'll go back here.
Yeah.
Yeah.
That's fine.
Okay, thank you. Okay, B right here. It's a little bit hard to tell from this particular angle. Anybody want to take a guess at what that is? A little knob right there. We've labeled it previously already in another diagram. Yeah, that's right, Tina. That is the pituitary gland. That's a terrible color to use. Pituitary gland. It works with the hypothalamus to help maintain homeostasis through hormones. C is part of your frontal lobe. You're just looking at it from the bottom. D is part of the temporal lobe. You cannot see the parietal lobe from this angle. It's all the way at the top, right? E, medulla oblongata. Now this next part is a little bit difficult to visualize without the actual model, okay? But you know, we're gonna cut away the cerebellum. We're gonna remove the entire brain stem, and then we're gonna take away the temporal lobe as well. And what you're left with is this. Okay, it's the same model, but we remove everything else that's blocking the way. And now you can see these structures, right? That's actually part of your lateral ventricles. Okay, very big. And you have two of them, of course. If you're wondering what these white lines are, those are actually one of your 12 pairs of cranial nerves. Okay, let's try this one more time. Which half of the brain is this? Okay, hopefully you will be able to tell this time. That is the left side. That's correct. That's the left side. Label functional area A and B. Okay, A here is the Broca's area, right? For articulate speech, right? Motor speech area. And we have B, which is the Wernicke's area for understanding speech and understanding written text, right? What is missing at question mark? Right. So you should have the cerebellum there. There is a groove right here, right? That divides the frontal lobe from the temporal lobe. That is called a lateral fissure. All right. So again, you will have to try to visualize this with me. This particular model, you can actually take A out and then take B out. Okay, and then we keep that thing where it is, and then it will look like this. Okay, do you understand? Right? You can remove the top and the bottom and pull them apart like that, and then you will be able to see the inside. Okay, so A is still the frontal lobe. C is the temporal lobe. Does anybody know what B is? B is referring to this lobe here. What do we call that? Not quite a midbrain. The midbrain is like down here. This is the lobe itself. There is a fifth lobe. Fifth lobe of the brain. Okay, that's where taste go. The insula. You might remember that. So here it is again, D. That is the lateral ventricle. Okay, so 1 second, I'll come back. In the previous picture, right, you see? That's the lateral ventricle viewed from below. And then you can see it here again. Okay. I keep on saying, if you stick your finger in to that gap under the corpus callosum, you can feel the lateral ventricle, right? So there it is. We are almost done the brain, guys. I know there's a lot of brain diagrams, but we're almost done. Okay, so here it is again. And we were just talking about the corpus callosum, which is right here. Okay, and again, like in a more accurate model, this would have been like an empty space. Okay, and you will be able to stick your hand in, and then you will be able to feel the lateral ventricle. Okay. But that's what $400 buys you. Not a very accurate model. Pineal gland. It was just mentioned in the lecture notes. I don't think I talked too much about it in the tutorial, but there's your pineal gland. The function of pineal gland is to secrete melatonin. What does melatonin help you do?
Sleep.
That's right, induce sleep. Okay. Yeah, your pineal gland is sensitive to light. If it's low light, it will secrete melatonin, and it will put you to sleep. That's why when I teach at school, I keep all the lights on, even though it makes it harder to see the screen. All right, so we have three things here. I think this is the last brain diagram, I promise you. 21, 22, and 23. Okay? In fact, we talked about this several times already. 21 is in front of the thalamus, so that's the third ventricle, or the thalamus, we can say thalamus. Okay, and third ventricle. Okay, the third ventricle is the space in front of it, right? 22 is below the thalamus, so hypothalamus. And you should know by now the thing that's dangling from the hypothalamus is the pituitary gland. Okay. Moving on. Label as many parts as you can from the spinal cord section here. Okay. So we did this before, right? Sensory information is gonna come in from the back. Right? There's gonna be an interneuron here in the gray matter, the butterfly. And then the action will get sent out from the front, right? So this is the afferent pathway. Right? Sensory information. And this is the efferent pathway, which is motor information. This thing here, that's like surrounding the spinal cord, is the meninges. This whole thing, same thing over here as well. That would be your spinal nerve. White matter on the outside, gray matter on the inside. See that tiny hole in the center? I'm not sure why it's not letting me draw the arrow, but there is this little hole in the center. That is the central canal. That's where the CSF is going to go down, right? Couple other things that you might want to label over here at the back. This area contains the ascending tract. Ascending tracts goes up to the brain, right? Sends the sensory information up. And at the front, that is where you will have the descending tract. Okay, when it comes back down. Okay, so that's everything we should know about the spinal cord. I identified two types of cells here. Well, this whole thing right here, right? That is the neuron. You can recognize the neuron because of the star-shaped looking structure. You can see the dendrites coming out, for example. These are like nuclei of glial cells, right? The supporting cells of the nervous system. All right, finally, we are done with the nervous system. Let's move on to the sensory organs. Okay. Let's see. Over here, A is the auditory canal. Auditory canal. B is referring to all these little tubes. Those are the semicircular canal. You have one that's parallel to each plane. And C, that's the Eustachian tube. And we know the function is to equalize pressure with surrounding. Okay, moving on here. Identify the anterior, lateral, and posterior. We can do that. Okay, the anterior one is this one. This is the anterior one. Right, it's going to be parallel to this plane, parallel to the wall that is on the side. The lateral one is this one. That's parallel to the ground or the ceiling. When you shake your head sideways, that's the one that will detect the motion. And the posterior one is the one here at the back, which is parallel to the wall in front of you or behind you. You tilt your head like that, you will stimulate the fluid, the cells at the base of this canal. All right, E is cochlea. Inside is where you will be able to detect sound. And all the information regarding, your body motion and the sound will be sent to the brain through D right here. That is the cochlea the other way around, vestibulo-cochlea nerve. Right? It's a mixed nerve. It contains two types of information. And next, what we're going to do is we're going to take this thing out and look at it. So there it is. X here, this thing with the big
Professor,
bulb. Yeah, go back.
last slide, we didn't identify the F.
Oh, sorry. Thank you for pointing that out. F is talking about this membrane here. That is the tympanic membrane. Thank you for reminding me. Okay, tympanic membrane. The eardrum, right? Now, when we take this thing out, we can see over here, right? Here it is again, the tympanic membrane. Let's just label that first. So on the medial side of the tympanic membrane, you will have this bone, right? The one that with the big knob. That is the malleus, right? Also known as the hammer, right? You have the pointy one. The pointy one, this one, that's the incus or anvil. The stapy is not on this model. Okay, I'm not sure if someone broke it off or whatever, but ever since I have this model, there was no stapy on it. Okay, but if there is a stapy, it will connect to right here, and it will look something like that. That would be the stapes. So collectively, XY and the stapy, right? It is called the ossicle. Ossicles. The function of the tympanic membrane is to convert sound energy into movements. Okay, so as it moves back and forth, as it vibrates the sound that it's picking up, it will cause the ossicles to move, right? And the sound is amplified that way, and then it gets transferred to the fluid inside the cochlea through the oval window. All right, here is a better picture. Okay. A is the stapes that was missing from the previous slide. Another name for stapes is stirrup. What does A connect to? So A, this surface, is going to bind to the oval window. On the cochlea. Okay, it's pressed on the oval window, so you can create a sound wave that travels inside the fluid. All right, here it is again, another ear. Label as many parts as you can. All right. Let's
Teacher,
yeah,
please, can you go back, please?
sure. There you go.
Yeah, thank you.
No problem. Okay, so label as many parts as you can. All right, I guess I'll just do that. And then you can tell me if I miss anything. Let's see if I can make it a little bit bigger, maybe. Okay. So here we go. These things, right? They are the semicircular canal, right? Semicircular canals. This circular structure cochlea. Connecting the two, connecting the cochlea to the semicircular canal, that would be the vestibule. The vestibule is responsible for detecting acceleration, right? Speeding up, slowing down in the car, falling, dropping. That's the vestibule. And that's the nerve that sends it all to the brain. The vestibulocochlear nerve. You can see the hammer here again, the malleus. You can see the pointy thing here. That is the anvil. And you can see the stay-piece here as well. Okay. I think those are the major ones that you can label. I mean, you can sort of see the tympanic membrane here, but not really. Is there anything anything else you guys want me to add? All right. I'm gonna do the eyeball next. So we have the 3 layers of eyeballs right here, starting at A. A is the sclera. It's the white part of the eye. That's where the muscle is going to be attaching to. B, that's the chloroid, is the vascularized layer of the eyeball. You can see all the blood vessels in the model, right? Along with the nerves. And then C, that is the retina. Okay. This eyeball can be opened up. You can see the line here. So I'm going to grab the top, remove it, and the inside is going to look like that. Okay. Can someone tell me what this crystal ball is? This little plastic circle sphere. E. What is that? No? That's okay. So this ball is the vitreous humor. Okay, that's the gel-like substance at the back of the eye to reinforce the shape of the eyeball. That's also the stuff that if there's too much pressure in it, it's going to press on your optic nerve and damage it, right? D here at the front, that is the lens. Function of E, function of the vitreous humor is to reinforce the shape of the eyeball. Okay. Next, we're going to remove the vitreous humor, the ball, and we're going to look at the model from the top. You can see at the back here, that is the optic nerve, okay, which takes the signal to the brain. We traced out the signal last time, right? With the optic nerve, optic chiasma, so on and so forth. We'll write it out again in a second. Where the optic nerve exits the eyeball, you're going to have a blind spot. That's what G is, blind spot. Okay, so stay the nervous pathway. So starting at the optic nerve, the information will go through the optic chiasma, depending whether the information from the right field or left field, you might cross the chiasma or you might continue on the same side. And then it will go through the optic tract, thalamus, and then finally arriving at the occipital lobe. Okay, there's a spot here on the side. That would be the fovea, right? This is where you will be able to produce a sharp image. That's the thing that you're focusing on, right? Whatever you're focusing on, that object will project an image onto the fovea. Eye is talking about all these muscle. It forms a ring, right? Remember I told you last time around the eye, those are the ciliary muscle, which controls the tension, how tight the suspensory ligaments are going to be. Which in turns control the thickness of your lens. Remember, if you want to look at far away, you need a thinner lens. For nearby, you need a thicker lens. Last eyeball. What is found in number 2? Okay. Might take you some time to look for number 2, but number 2 is right here. Okay, this is number 2. So what's found in number 2? Aqueous humor, which nourish the lens. The lens as well as the cornea. The lens and the cornea. Okay. Oh, I think I made a mistake earlier. I just realized it. Sorry. This is not the lens. This is the cornea. Just a couple of slides before. This is the cornea. Sorry about that. The lens would be like behind it. The first part that the light strikes before entering your eyes would be the cornea. Okay. Clouding of J. What do we call it when this thing turns cloudy? Anybody remember? Turns white, and then slowly you might lose vision. Cataracts. All right, moving on. This is everything. Okay, let's see what label all the numbered structures. Okay, so over here, that is the cerebrum. Cerebrum. Number 5. The sharper color, can't really see them. Number 5 is this thing. You should know what that is by now, right? We've labeled this at least 4 times today. corpus, callosum, communication between the two hemispheres. Number 9 here is nasal cavity. Seven is pons. Six is cerebellum at the back. Now this is the base of the skull. Okay, so by the time you get out here, number 8, that is already the spinal cord. People usually have trouble with number 10 because they're not used to seeing it like this. But number 10 is actually your tongue. Okay, very big. (11) and 12, I don't have room there. I'm gonna write here. Earlier today we did fill in the blanks, right? The hard palate is made up of 2 bones. So 11 is the maxilla, whereas 12 is the palatine. And the extension. Oh, sorry, my bad. 11 is the hard palate. 11 is the hard palate, which is made up of the 2 bones, the maxilla at the front and the palatine at the back. Okay. And 12, by the time you get to 12, that would be the soft palate. If you curl your tongue and then you feel the top towards the back, it no longer feels hard, right? It's just like tissue. That's the soft palate. And the extension of the soft palate is the uvula. Okay. Usually people have trouble with this one as well. That is the adenoid tonsil. And that's the one that gets like when you have a cold, right? And you have like runny nose, that's the one that gets infected. Okay, I think that's everything. Moving on. I know this is like for the earlier units, but it's like a whole body model, right? So we just want to be thorough. 16. 16 is the thyroid cartilage. (17) here is talking about that gland. That is the thyroid gland, which secretes hormones for regulating metabolism. 19, 20, 21. 19 is superior lobe of the lung. 20 is the middle lobe. And then 21 is the inferior lobe. Okay. You have 3 lobes on the right side, only 2 lobes on the left. Right here, it's hard to see. That is the aorta, right, coming out from the heart. Okay, I think there's another number here, number 24. That would be the pulmonary artery. Okay. All right, we're almost there. Just hanging tight. And now we're the abdomen. Right? What is 35? This is the digestive system now. Do you know what 35 is? Anybody? The stomach is normally right here, okay? And behind the 35, yeah, that's correct, Tina. That is the pancreas. 41, that's the transverse colon. 34 here, that would be the second part of the small intestine, the jejunum, which is for nutrient absorption. And there's one thing that's over here. You can't really see it, but it's 42. That one right there, that's the descending colon. If you think about the diagram, right? Large intestine begins with the cecum, and then you have the ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and anus, right? Okay, I think we have a diagram coming up to label those as well. Not sure why we have two of the same slide. Move on. Familiarize yourself with different parts of large intestine. That's basically what I just explained to you, right? Ascending, transverse, descending. And then you have the sigmoid, rectum, and anus. Okay, so just study this diagram, make sure you understand it. Okay, here is the stomach. A, that's the esophagus. Over here is a sphincter. You should know that by now. That's the cardiac sphincter. C is the actual stomach. And you can see all these foldings, right? The foldings, they are called rugae. Down here, we have the pyloric sphincter. And this last part here, that's the first part of the small intestine, the duodenum. Okay, so the pyloric sphincter will control how much of the chyme is allowed to go into the duodenum. Okay, A here, that's the liver. B is the gallbladder. C is the pancreas. And D, we just label it, is the duodenum. Earlier today, we already stated the function of pancreas, right? It's to produce the sodium bicarbonate for neutralizing the chyme, as well as to produce digestive enzymes. So I'm not going to write that again. Okay, this is an esophagus slide. That's how big it is. Identify the 4 layers of the GI tract. Okay. So the outermost layer, let me try to highlight it for you. It's a little bit difficult to do, but this very thin layer at the edge, that would be the serosa. Okay. These 2 layers from here to here, that's the muscularis. Okay, you can see the circular muscle and the inside and the longitudinal on the outside. Okay, so starting from like here all the way to the outside, that's the muscularis. Muscularis. Okay. The submucosa is the one that's stained like a light blue right here. Okay, so this region, that is the submucosa. Okay, you see the submucosa here, and some part it's like, you know, deeper than others. And finally, the mucosa is just the innermost layer. Okay, it's staying like dark purple here, right? Okay, it goes all the way around. That's the mucosa. Okay, and you can always find these 4 layers when you look at the GI tract. And let's look at it. This would be like a cross section, right? Transverse section. Let's look at it from a different perspective. This is the small intestine. And let's see if we can find the 4 layers again. Okay. So over here, we have the serosa, followed by the muscularis. And then here we have the submucosa. And the mucosa. Okay. Just 2 more slides. Identify the following. Okay. So the label looks a little bit weird because we didn't learn it using those names. Those are the Latin names, which is more difficult to remember. So PC is talking about this whole thing. And we call it the circular folds. When we were doing the study guide earlier, right? Remember, draw it out like that, right? That's the circular folds in the small intestine. The ME, which is right there, that is talking about the muscularis. Muscularis. So from the circular folds, you have these things that are coming out, right? That's the V, which is the villus, or the villi for plural. In the center of the villus, you will find the lacteal, which helps with the fat absorption. Sub M is talking about the submucosa. And this layer here, that's the mucosa. Okay, you can try to match this diagram with the previous one here. You should be able to see the resemblance of the different layers. Okay, the final slide. Which side is the surface of the tongue? This is the surface. Okay, you would know because it was in the PowerPoint. There is a diagram for that. And these little comb-looking thing, each one of them is called a papilla. So there's a papilla, and on the side of the papilla, right, these little oval thing that looks a little bit pale pink, right? Those are your taste buds. Okay, that's where you will detect the sweetness, the sour, and so on and so forth. All right, that's it. Okay, well, thank you for sticking around. It was a very long session, but thank you for staying. So you probably have read the announcement. The schedule was changed for the remainder of the semester. So next class will be on the Thursday. Okay, it will be next Thursday instead of the Tuesday that was being planned originally. So on Thursday, we'll do a test 2 review. I'll post the review sheet. And then instead of having the test on Thursday, I will let you have access to the test next Thursday after our tutorial, and then you would have until Friday to finish it. Okay. Originally we planned to do it on Tuesday, and then you can write the test on Thursday, but because of the scheduling conflict, we will have the review session next Thursday, and then you can write the test anywhere between Thursday 3:30 to Friday midnight. All right. So I'll post the announcement, and then I will give you the link for the lab test as well, right? Lab test for this particular lab. And yeah, okay. Thanks again. I'll see you next Thursday then. Have a good one, guys.
Teacher, professor,
Yes. Yeah. Go
when
ahead.
the lab quiz will be due?
Lab quiz. I think it's gonna be due next Wednesday. I have to double check. Okay, hold on a second. Let me just quickly pull that up. That would be lab quiz number 2 is due on the 15th. 15th, so that would be next Tuesday.
When it will be available?
It should be available now already. Yeah, it says available on November 8th, so you should have access to it already.
Okay. Okay. Thank you.
Okay, have a good one, guys. Bye.
Bye.